Hello everyone, this is Mrs. Gabrick and I'm bringing you the first of several videos on genetic problems. Today we're going to learn how to set up and solve genetic problems. So this is going to be a very important video because from here on out, every problem we do, if you have the basics down, it'll be very easy. So I'm going to ask that you turn in your packet to the page that says how to set up and solve genetic problems. And there you'll be able to follow through the problems that I'm doing here on the video screen. I'll also put this up on SharePoint, so if you want this PowerPoint, you'll be able to access it there too. Alright, so hopefully you found that page in your packet, how to set up and solve genetic problems. If you're having trouble, pause the video and find that page. Alright, so the first problem we're going to use is about hamsters. In hamsters, black coats are dominant to white coats. By dominant, what we mean is, of the two alleles that code for coat color, black will always dominate white. So if you have both alleles, a black allele and a white allele, you'll see the black allele. Black will be expressed. Remember, for each gene, we're going to have two copies, one on each homologous chromosome, and those copies are called alleles. So we're going to cross a homozygous black-coated hamster with a white hamster. Homozygous, homo means the same, it means the alleles of the black hamster are the same. So in other words, he's going to have two black alleles. And we're going to cross him with a white hamster. Since white is recessive, the white hamster must have two alleles for white. If the white hamster had a black allele, it wouldn't be white, it would be black. When we do these problems, we're going to want to determine the genetic and phenotypic ratios for both the F1 and the F2. The genotypic ratio tells us what alleles they have. Are they big B, little b, or little b, little b? The phenotypic ratio tells us what they look like. Phenotype is, are they black or are they white? But just knowing the color may or may not tell us what their genotype is. So, step 1. Determine the genotypes of the P generation. The P generation is the pure parental generation. They must both be homozygous to be in the P generation. Since black is dominant to white, we're going to use big B to represent the black allele. And then we'll use little b to represent the white allele. Remember, our hamster that's black was homozygous black. That means he's going to be big B. And we're crossing him with our cute little white hamster. Because white is recessive, our white hamster must have two little b's. This is our P generation, so I put the P to the left. Because we're crossing them, which means we're breeding them, we use an X to represent that. So we're crossing a big B hamster crossed with a little b hamster. Step 2. Determine what type of gametes or sex cells the organism can make. It's a good idea to circle your gametes. That keeps them or distinguishes them from the genotype. So because my homozygous black hamster has two big B's, he will only make big B sex cells. This goes back to meiosis, and this is called Mendel's law of segregation. Each big B is on a different homologous chromosome. So we separate the homologous chromosomes in meiosis. So I'm going to put a big B in one sex cell, a big B in the other sex cell. With the white hamster, same thing. The two little b's, the two alleles, are on homologous chromosomes. We will separate them into separate sex cells. Step 3. Look at the possible combinations for the offspring. In this case, there's only one. These are the little hamsters, and as you can already see, even though they're very tiny, they're all black. The reason for this is they got a sex cell from the black hamster that said big B. They got a sex cell from the white hamster that had a little b allele. So all of these hamsters are going to be big B, little b. That's their genotypic ratio. They're all black. That's their phenotypic ratio. Now, if all of them have the same genotypic and phenotypic ratio, we are okay with you saying all big B, little b, or all black. If you prefer to write it as a ratio, you can do that. You would have to say, oh, there are 0 big B, to 1 big B, little b, to 0 little b. Or you could have 0 big B to 4 big B, little b, to 0 little b. We will always reduce, but you don't have to. For the phenotype, you could say 1 black to 0 white. Or 4 black to 0 white, in this case 5 black to 0 white. Okay? But again, it may just be easiest to say all black. That's my F1, the first generation. These are the children of the black and the white hamster. For our next problem, we're going to find the F2. That's the kids of the kids. And yes, in this case, it will be ancestral. We are going to take 2 of these little black hamsters, once they've grown up, male black and a female black, and cross them. So there's my F1 now. These 2 hamsters here are both my F1s. They've grown up a little bit. And we're going to take the F1 and cross them. Remember, their genotype was big B, little b. So if we cross big B, little b with big B, little b, we're going to get the F2. We need to determine what type of gametes the F1 can make. And again, it's still a good idea to keep those gametes circled. Now, again, using Mendel's law of segregation, we're going to separate our 2 alleles. Remember, they're on separate homologous chromosomes. So I can put a big B in one sex cell, a little b in the other sex cell. The same for our other hamster. Big B in one sex cell, little b in the other. We always segregate our homologous chromosomes. Now, step 6, for this one, it's a little bit more complicated. We're going to look at possible combinations for the offspring. And since we have more possibilities, a Punnett square is a good idea. So to do this, you're going to put one parent's gametes on the top and the other parent's gametes on the side. There we go. So I put Dad's big B sex cell on the top and his little b sex cell on the top. I put Mom's big B sex cell on the side and her little b sex cell on the side. And then in the box, we can determine the possible combinations. So in the first box, if I get a big B sex cell from Mom and a big B from Dad, my kid is going to be big B, little b. If Mom gives this kid a big B and Dad gives the kid a little b, I'm going to be big B, little b. Here, we got a big B from Dad and a little b from Mom. You'll notice, I always keep my big B's first. I just find I make less careless errors, but it's not necessary. If you put little b, big B, it's the same thing. I just find I make fewer careless errors. And our last possibility is we could get a big B from Mom and a little b from Dad. Step 7, write down the ratios for the F2. If you go back and look at that Punnett square, you'll notice we had some black hamsters and some little white ones. Alright? So our F2, the children of the F1, our genotypic ratio would be 1 big B to 2 big B, little b to 1 little b, little b. Our phenotypic ratio, because big B will be black and big B, little b will be black, we're going to have 3 black hamsters to 1 white hamster. That's how you set up a genetic problem. Now, we've got a new problem that we can do here, and if you turn the page to the back, you'll see that there's another problem. In this problem, we're looking at dogs. So step 1, in this problem, we've got floppy ears, big F, are dominant to upright ears, little f. We want to figure out the expected genotype and phenotype ratio for their offspring. In this case, I'm starting with a heterozygous floppy dog and an upright-eared dog. This is not quite the same problem because we're not starting with a pure generation. It's a heterozygous floppy dog, meaning it's not going to be big F, big F. Because it's heterozygous, it's big F, little f. And our upright dog, because that's recessive, it's going to be little f, little f. Step 2, what type of sex cells can these dogs make? Again, circle your gametes. Big F, little f, we're going to separate our homologous chromosomes, and that dog can make sex cells that are big F or sex cells that have a little f. The straight-eared dog can only make little f sex cells. Again, step 3, good idea to do a Punnett square. I'm going to put the heterozygous dog's sex cells across the top, the straight-eared dog's sex cells on the side. Look at what possible combinations, and what I would suggest is maybe pause this, see if you can figure out what goes in each box, and then check it. Okay, let's see if you got it right. Big F, little f in that box, little f, big F, little f in that box, and little f, little f. Can you figure out the genotypic and phenotypic ratio from this? Maybe pause it again, and then when you think you've got it figured out, check and see if you got it correct. Alright, did you do it right? The genotypic ratio was 2 big F, little f's to 2 little f, little f's. Even though I didn't do it, if you want, you can put in the 0 big F, big F. But we're not going to make you do it if it's 0. The phenotypic ratio is going to be 2 floppy to 2 straight. If you would like to reduce these, you could say 1 big F, little f to 1 little f, little f. I always was taught to reduce it, but it's not necessary. But you may see on a quiz that the answers, or on a multiple choice test, that the answers will be written in a reduced form. So you could also write this as 1 floppy to 1 straight-eared dog. Heterozygous. Remember we talked about homozygous means they're the same? Well, heterozygous means you have one of each allele, and you know how we enjoy jokes in honors biology. Heterozygotes. They're just a little uneven. Alright, so we'll see you tomorrow in class, and during class we'll let you practice some problems and work on them individually and with your groups. See you tomorrow.